Thank you very much, Matt, for the kind introduction. And Julie and I also we're really excited to be here. It's our first time here. It's been a really fantastic, inspiring, and sometimes surprising 2 days. We hope to share a story with you that will resonate with a lot of the themes that have already come up in the conference so far. We're going to do this as a little bit of a tag team, but Julie and I have decided that I'm going to help get us kicked off by framing the story just a little bit. For those of you who don't know me, I'm a neurologist and a geneticist at Boston Children's Hospital. I have the privilege of working with a group of talented folks that have been working with me to study topics listed on this board. We do a lot of human genetics, we do a lot of gene discovery, and we focus a lot on rare disease, especially rare neurologic disease, themes that we've been talking about in this session and this morning too. And along the way, it's really dawned on us how fortunate we are to be doing this work at this time. We live in the age, as we heard about this morning, of the $1,000 genome. The first genome was completed in 2003 at a societal cost of $3 billion. Now folks can ship off a sample of blood or saliva and for $1,000 have their complete sequence delivered to them. And as a consequence of that technological advance, genome sequencing has hugely accelerated the diagnosis of patients with rare diseases. And you heard from Lynn through the UDN that with a lot of hard work, blood, sweat, and tears, we can now achieve diagnoses for up to a quarter of these patients. A quarter, still, there's room for improvement, but there's unprecedented new gene discovery and diagnosis available to us with the tools that we've got. The challenge, though, in this new age is how do you actually turn these diagnoses into treatments? And developing drugs still takes a long time. And what are paradigms that we need to be thinking about to accelerate that process? So with that slightly long-winded intro, in contrast to many of the types of gene discovery projects that Lynn and others in this space and my lab works on, where you analyze tens and thousands or hundreds of thousands of patient exomes and genomes to solve cases, Julie and I are going to tell you about one case. And the particular conceit that we're going to use for this presentation is the format of a traditional medical grand rounds. So imagine yourself from this beautiful auditorium now transported into an old school medical operating theater. And I have the privilege of presenting a family to you and telling their story. That patient, her name is Mila, and rather than me tell you about it, I'm going to hand that over to Julia.
I've spoken with Tim a million times now around the country, but I can never start without crying. I last was in this auditorium 10 years ago, before I had children. I don't think I ever would have imagined that I would be here today speaking about my own daughter and about rare, fatal genetic diseases. My daughter Mila was born 8 years ago here in Boulder, which is where I call home. And she was a very typical little Boulder child. She was very active. She was hiking for hours on end when she was 1.5 years, 2 years old. She was skiing by the time she was 2. We would drive up the canyon, and it was my dream since I moved here as a avid skier to bring my first child to go skiing. And I did that for years with her. She was incredibly social. There's some people here whose children grew up with Mila. And she was incredibly outgoing. And when she was about 4 or 5 years old, she started to have a number of different problems, of which many kind of were under the radar. People told me it was kind of nuts because she was so advanced verbally, When she would climb rock walls, when she was 2 years old, everyone was like, Julia, she's fine. But she started stuttering, and then I noticed that the stuttering therapist thought that it was a quite unusual type of stutter. She started stepping on all of our books at home, and we had half of our little children's books were broken, and I realized she wasn't seeing them because she was just walking over them. Toys were broken that she would step on. She was pulling little objects in really close. And I carried around a little piece of paper with me where I'd scribbled neurological conditions, ? I didn't even know what a neurological condition was, except for that it possibly came from the brain. And I started with stuttering. And I started adding walking because she was tripping eventually by the time she was 5. Vision came up as another one. And I had this little kind of scrap piece of paper that I would carry around with me. And I started bringing her to doctors, not only around the Colorado area, but all over the country, into Canada, the East Coast, the West Coast. I was lugging around my little infant son at the same time, and just no one could figure it out. And I would go to an ophthalmologist, and they would be like, I don't know, I think she's fine. And I would say, but look at my piece of paper. She has other neurological symptoms. And I would go to the orthopedic doctor, and they would say her feet are interned, but they'll eventually fix themselves. And I would say, but what about the other neurological symptoms? At some point, I gave up, and I noticed that there was a week in November of 2016 where I had gone on a few runs, and I just came back crying every time. And I just thought, something is just so wrong here. I don't know what it is. And so I just packed a duffel bag, and I put my daughter in the car and drove to Children's Hospital Colorado ER. And I just thought, there's no way they can release her because there's just too many things going on. And I was correct. We spent a horrible week, where she underwent 1 million tests. And by the end, I was sitting next to my husband with 15 doctors in white coats with very solemn faces, with their heads down, saying, I'm so sorry, but we don't have the genetic results back, but your daughter has one of these 2 conditions, and neither of them are looking very good. So I left the hospital feeling so devastated, but also having no answer after years of wondering what was going on. Eventually, I got a call a week later and was told that she had Batten disease, which is a rare neurologic condition. It's fatal, 100%. There were no treatments. I asked everyone, there has to be something, there has to be a pill, there has to be some therapy that helps. And the answer was no, there's nothing you can do. You can prepare to watch your child lose all her abilities and have her die in the next few years. And by the way, your son might have this too, because he is 2 years old and he's too young to know. This is before Mila had symptoms. And they didn't give me much hope, but there really wasn't much hope at that time. So I began to call scientists around the world. Every article I could read, possibly get my hands on Batten disease, I would call or email the scientist and bother them until they finally spoke with me. I asked them all, is there any reason for me to have any hope? They all told me, I'm so sorry. This is one of the worst diseases I've ever come across. We're probably 10 years away from a treatment. But they did say one thing, which is what I held on to, which was there's some reason to believe that we might be able to fix genetic conditions like this, but we don't know when that's going to be. And that's all I needed to hear. I figured out pretty quickly that I needed to start a foundation. I didn't even really know what a foundation was, to be honest. I just called anyone that I knew that had a foundation and asked them what it was and how I could start one. And I started one, and I quickly figured out that gene therapy, traditional gene therapy, kind of came up as the only bet I had, and that the price tag was $4 million. I unfortunately made a calculation one day of how much I would have to raise a day in 1 year to get to a gene therapy, and that was not a good week. I let that go and tried not to think about it, and I just decided that the only way I was going to raise that money and try to get a treatment for my daughter was to just tell my daughter's story. And so I just opened my doors and let anyone, press, news channels were showing up at the time when I just wanted to crawl in my closet and cry. But I let them in, and I started to tell her story on the internet. And thanks to some really wonderful friends here who worked for a large international marketing and advertising company, they so kindly didn't ask anything of me. They just said, look, we're going to show up at your house in a few days with a camera, film crew. We're going to make you a video. And thank God they did that, because the video that I made has been my business card that has opened the doors to countless physicians and scientists, researchers, and there's nothing more powerful than a video. You can say all that you want in an email, but it just doesn't really tell the story correctly. So they made this video, which I just would love to show. It's just 5 minutes. I think it's something that Tim and I show at every meeting, every scientific meeting, meeting with the FDA, meeting with anyone. We show it because it's really important to set the stage of rare disease and what we're trying to do and how we're trying to help other children. So I think we'll start just by watching this 5-minute video.
You make me happy when skies are gray.
So our daughter's name is Mila Virginia Makovitz. She was born on November 5th, 2010. When I think about Mila as an infant, I think about smiles and laughter and health and...
Half an hour after she was born, she was strong enough to hold on to my finger and I could almost pick her up off the bed.
When Mila was a toddler, she was a climber, she was a hiker, she was a runner.
How old are you, Mila? Here's our sugar high daughter.
Slide, slide, slide. Woohoo!
Very physical, always loved to ride a strider,
Nice, bug.
play in the snow, go sledding.
Coming!
We noticed that Mila was getting stuck on her words. She would say, Mommy, Mommy, I have to, Mommy, I have to, and not be able to continue. And then suddenly, around 3 years old, we started noticing that her feet were slightly unturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way, and then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. We took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She could no longer see anything. What's brought her so much happiness is playing with her toys and or looking at her books and watching TV and playing with her brother, and suddenly it looked like she could no longer play with anything. It's so hard to see such an outgoing little girl not be able to play with other kids and not be able to play with toys and not be able to watch Elmo anymore. Children with Batten disease are destined to lose their vision, their ability to walk, their ability to talk. They lose their ability to swallow, and they eventually become bedridden, and they don't usually live until much after 10, 11, 12 years old.
It's such a rare disease that all the doctors that you see have never experienced it before.
When Mila was born, it was the happiest day of my life. It gave my life purpose, and I just asked myself all the time, how is it possible that we have such a wonderful life? And then we had our son.
Aslan, he's 2.5, but he doesn't show any sign of being affected, but we know that there is a chance that he is.
The night before we took Mila into the hospital, he looked at me at the dinner table and said, "Mommy, is Mila gonna be okay?" Our beautiful family and life seems like it's slipping away every day. Her smile and her laughter and her spirit is the same. If we want to save Mila's life and the life of other children who are suffering from this horrible disease, we need to raise $4 million right now to get a clinical trial up and running. We want to do everything that we can to stop other families from experiencing the pain that we're going through right now.
You never know, do you, how much I love you. Please, please don't take my sunshine away.
Oh. So I'll let Tim take over from here, but this video has been incredible help to me. They've had 2 million views in the last 2 years. And when I approach scientists, doctors, donors, many of them have already actually seen this video. So I didn't know what it was like for something to go viral, but I can tell you that it greatly helped allow me to find a treatment for Mila. I'll let you take over, Tim, from here.
So this is the disease that Mila's medical team in Colorado had given her a diagnosis for. It's called Batten disease, and it was picked up, the critical test was an abnormal skin biopsy, a small snip of skin from Mila that showed abnormal patterns of inclusions in these parts of cells called lysosomes, which are, in a way, the recycling waste containers of cells. And what happens in Batten disease is actually caused by mutations in one of 14 different genes. And when you knock out both copies of any one of these 14 genes, all of these are critically involved in lysosomal biology. And unfortunately, what happens is that these recycling centers clog up with material that you can see on a microscope that eventually makes the cells sick and eventually kills them. And this affects the brain and the eye, and it explained the symptoms that Mila had been experiencing. Her particular subtype was called CLN7, the 7th gene in this series, and it's a rare subtype of a rare disease. It's an ultra-rare disease. There are only 70 cases reported in the literature, and currently there are probably fewer than 20 active cases in the world that we know of today. And the age of onset was typically around 3.5 years. And the symptoms progressed from vision loss, seizures, motor and cognitive decline, and unfortunately to death. Typically, age of death around age 11. Now, Mila's family had worked hard to recover from the diagnosis amazingly quickly and launch an effort to start a gene therapy. But there was a wrinkle, and the wrinkle was that actually at the molecular level, she only had half a diagnosis. She had cytopathology, microscopy showing that this was likely Batten disease. But at the genetic level, you need 2 mutations to prove a diagnosis of Batten disease. And the team could only find one. Dr. Larson had correctly explained to the family that this happens sometimes in clinical diagnostic testing. About 10% of the time, we find one mutation and we have trouble finding the other. And in explaining why this is the case, it's that sometimes mutations that we think are present are actually part of the dark matter of the genome. They're parts of the genome that we actually can't see. And that's because 99% of the genome is actually not included in clinical gene testing. And you might think, well, that doesn't sound like a very good idea, but it actually has some normal basis, some logical basis to it. Here's a gene, a typical gene that has these different gray boxes, which represent the actual important parts of the gene that code for the instruction set to assemble this gene. And typically, clinical labs target these gray boxes, which are called exons. These are the coding instruction sets. But exons only constitute 1% of the genome, and it turns out that rarely, occasionally, there's important stuff in these yellow regions here. And the only way to get at these, and the only way to complete a diagnosis for Mila, is to do whole genome sequencing to get all of these parts. And so it was because of this situation that I fortuitously got involved in the situation. Julia actually began to ask around to find what clinical lab in 2017 could offer whole genome sequencing for her daughter, because she was determined to find the second mutation. The second mutation was critical because even if she could launch a gene therapy, would she even qualify for the trial if we weren't 100% sure that this was the right gene? Moreover, there was Aslan, Mila's little brother. And how would we be able to clear him from the diagnosis if we didn't know exactly what was going on with Mila? And so Friday night, January 13, 2017, my wife turned to me and showed me this Facebook post, which had been put up on a group called PMG. And PMG stands for the Physician Moms Group. Turns out that my wife is a physician too, and we have 3 young girls. And she passed this message along, and it said, "I'm hoping the power of this group can help my dear friend. Julia and I were college best friends. Her 6-year-old daughter has just been diagnosed with Batten disease. They have localized one of the defects, but need the second. Their doctors are recommending whole genome sequencing. If anyone can help in any way, please. They would appreciate it so much." And our research group has had the experience of doing a lot of whole genome sequencing over the years. Actually, we were lucky to be one of the very first to do it, beginning around 2009 or so. And I spoke to my wife about it, I spoke to my team, and we decided that here's a case that we ought to try to help. So that very next day, I called up Julia, and we had a phone conversation. And I said, we have experience in this space. Oh, and I forgot to mention, we had actually at that point just co-founded a company that does clinical diagnostic genome sequencing. And we offered to help solve the case. 4 days later, we had consented her to a research protocol at Boston Children's. A week after that, we had blood samples shipped. And a month after that, we had completed whole genome sequencing of Mila, mom, dad, and her little brother. 10 days later, we found something rather unexpected. If folks haven't seen whole genome sequencing, this is what it looks like from a bird's eye view. When we sequence genomes, we actually shotgun blast them into tiny little fragments of genetic code. Each is about 150 strings of A's, C's, G's, and T's. And all of these then get lined up against a reference sequence, that 2003 reference map of the human genome. And each of these little gray bars represents a little read. And this was Mila's sequence, mom's sequence, dad's sequence. And what we immediately found was the one mutation that Dr. Larson here in Colorado had already found. That was the known pathogenic mutation, and we found that it came from dad, and that meant that all we had to do was now look for something that came from mom. Well, when we turn now to look at mom's sequence, we notice something unusual. And this is something that's actually quite subtle and that most clinical diagnostic labs in 2017 were not going to pick up. And it explained why perhaps it had been missed. And what we saw was a cluster of colored reads here that were present in Mila and in mom. And I'm being mysterious because I haven't explained to you what color means. When you zoom in on these colors actually indicated that these were reads that didn't perfectly line up in the way that you expect them to the human genome. They were actually chimeric reads, chimera. Well, so half of the reads lined up properly, and then the second half was from someplace foreign that didn't belong at that site. There were 2 types of chimeric reads, some that lined up like this and then switched into foreign sequences of just T's, and another family that lined up like this in the opposite direction and then switched into a sequence of a repeating 6-letter code. And after a lot of puzzling through and staring at this, here's the poly-Ts and these here, and here are the tandem 6-letter codes here. We realized that this actually represented an insertion of extra DNA that didn't belong at this site. And all in all, putting a long story together and compressing it, we found that there was 2,000 bases of extra DNA inserted into this site. Where did it come from? It turns out this is a sort of an interest, but not important to the story. This is a 2,000 base sequence called an SVA sequence. This has the technical term of being called a retrotransposon. You can think of it as a sequence of jumping DNA. And it turns out that retrotransposons and SVAs are a type of genomic virus that actually live in our genomes, and they copy and paste themselves hither and thither. Luckily, not very often, but once in a while they can pick up and do this. And if they do this jump or copy-paste, they can cause a disease if it happens to land in the wrong spot. So that's what we had found, and we had to ask, well, what's this insertion actually doing to the gene? There is a stretch of extra sequence here, and we hypothesized that it actually might be changing the assembly of the gene instruction set. And our hypothesis was tested by a number of different experimental methods that I won't go into, and essentially proved to be true, that this insertion of foreign DNA was causing the normal exon 6, instruction block number 6 of this critical gene to now splice and be connected to a region of DNA that actually was nonsensical. It didn't code for anything. And it actually encoded a strong stop signal that rendered the cell unable to read the rest of the instruction set downstream. This was the answer. It cut the gene in half and solved the mystery of what the second mutation was. Pass this to you.
So at some point in the spring of 2017, we got a call from Tim, and we had been working on a gene therapy and just hoping for the best, but learning that gene therapy took a lot of money and a lot of time, and that it was probably going to take us years and very likely not be in time for Mila. And we got a call from Tim, and he said, we found the missing mutation, and your son does not have Batten disease. I can't quite describe that day. I'd been looking at my son in his crib every night for 4 months, thinking it's possible he's going to lose all the abilities that Mila's losing. So that was very, very good news. And then he paused and he said, and I have an idea of how I might be able to help your daughter. And I will never forget that moment, because I just thought, how is this possible? I've been told that we're 10 years away from any treatment for my daughter. I don't think that the gene therapy I'm working on is going to be in time. And I had only hoped that this lab would just help me find the missing mutation. I never dreamed that that might turn into an actual treatment. And so from there, I'm going to let Tim tell the details maybe of what happened after that turning point that went from diagnosis to a possible new treatment.
So the idea that Julia was alluding to was that we realized, looking at this particular mutation, and we're missing a couple of blocks here, I don't know what happened to the graphics, but looking at this particular mutation, here is a situation where actually all of the coding instructions for the CLN7 gene were intact. They were present, but their usage was just being corrupted by this interloper, this new foreign DNA element that was coming in here and mucking things up. And so the very simple idea was, could we use a type of a drug called this to block this abnormal assembly event? And if we could now tell the cell to ignore this interloping DNA, could we now just, with the cell now, assemble the gene in the proper manner and restore a normal gene function? The type of drug that we were thinking about was something called an antisense oligonucleotide. And I have to explain that a little bit. We only thought about this idea because just the month before Julia's family reached out to me, we were reading first in the New York Times and then in the New England Journal, sometimes it works out that way, that there was this amazing new clinical result with a drug called an antisense oligonucleotide. Antisense oligonucleotides were invented, in a way, here in Colorado by Marv Carruthers, who designed the chemistry necessary to build synthetic DNA out of chemical building blocks. And what this study had shown was that properly arranged, a series of synthetic DNA bases connected together and targeted to the genomes of patients with this devastating...
Here's what I found.
Here's what we found. That an antisense oligonucleotide could be used to remarkably reverse the clinical deterioration of this disease called spinal muscular atrophy. And this is a really tough disease where infants are born really weak, and they just get progressively weaker and weaker, and eventually they're on respirators and they can't survive. This particular drug worked by changing the assembly of one of the genes that's critical to the survival of neurons that support breathing and other motor function. And it worked in exactly the same way that we were looking and considering developing a drug for Mila. And there were just too many close parallels to Mila's case for us to ignore this. This is an autosomal recessive genetic condition due to mutations in one gene. So was Mila's. This is associated with brain central nervous system degeneration. Unfortunately, so was Mila's disease. And they used this particular chemistry with an antisense oligonucleotide. Could we do the same thing? They gave this drug to infants with SMA via spinal tap. Could we do the same thing? This drug was able to activate a latent gene by changing the splicing. Could we do the same thing? And so we began pulling all the papers that we could that described how the drug for SMA was designed. And we went back 10 years to the very first proof of concept in human cell lines, and moving on to the proof of concept in a mouse, pharmacology in mice and monkeys, phase 1 studies, phase 2 studies, phase 3 studies. And I looked at my team and I said, okay, you guys can have 6 months. The only benefit that we had, of course, is that we could do this. We had a template to follow. So we developed the following scheme, and the idea was to try to develop an N-of-1 customized therapy just for Mila. And again, the idea was to do a patient-specific screen. We would computationally identify regions around this mutation that we should target with antisense oligonucleotide drugs. We would then design those, and then we would ask nicely for cells from Mila and her parents, skin cells that we could grow in a dish, and then use these skin cells to test these drugs and try and get normal gene assembly restored to those cells. And we quickly found that we were able to get this to work. Here's one of our very first experiments doing this. We designed a series of antisense oligonucleotides here. Here are patients' skin cells from Mila. And we developed an assay where you see these two bands here. There's a top band which represents the normal CLN7 gene product, and then a bottom band which represents what happens when the mutation corrupts it and it misassembles. And you see that there's about a 1-to-2 ratio between what's normal and what's mutant. After administration of one of our drugs, we were seeing that this ratio was now flipped. This top band here is much brighter than the lower band. And that was just instant visual gratification that we were on the right track. We, of course, repeated this many times. We tried it in different isolates of her skin cells. We tried it in her blood cells. We did dose response curves. And everything that we did led us to think that we were doing pretty well. This is a dose response curve showing the potency of this drug. And if you're not a pharmacologist, these numbers may not mean much to you. But the take-home message is that the potency of this drug to boost levels of normal splicing and suppress levels of abnormal splicing was right around the range of what the scientists had been seeing at this stage of study for the development of a Spinraza drug. We were doing just as well as the published literature. And we did RNA-seq transcriptome experiments to confirm that this was actually working on a genome-wide level. And we could see that with controls there was only around 10% normal splicing happening at Mila's gene locus, and that we could boost this by about 3 or 4 times by giving our antisense oligo. Well, that's RNA splicing. What about function? Are the cells any better? Well, here it turned out that we had a wonderful colleague at Northwestern who had been studying the cell biology of lysosomes in diseases like this for quite some time. And he helped us with an experiment that allowed us to visualize this. Here is a normal cell, electron microscopy of a normal cell, and here's an electron microscopy of Mila cells, and you see all these white bubbles that actually represent the duplication of lysosomes that are frantically trying to recover from the genetic insult that they've experienced. And we saw that the number of these was hugely abnormal, but that if we added our oligonucleotide, we could get this number to be reduced hugely, both in number of lysosomes and size of lysosomes. So sometimes almost so that they were entirely back to normal. Sometimes they were just a few more than normal. We did many other experiments that I'm not going to go through. This slide is just to cow you all in the submission that we were very careful and convinced ourselves via a number of assays that Joe Mazzulli set up that our ASOs seemed to be working, our drugs seemed to be working. And actually, I had a hard time believing initially how well this seemed to rescue, but it really did seem to do that. So let me quickly recap. So in December was Mila's clinical diagnosis, the first mutation identified. In January, we first came in contact with the family. In February, we had finished whole genome sequencing. In March, we had identified this splice-disrupting retrotransposon as the second mutation. In April, we began to talk to Julia and Alec, saying, we might have something here. You know, don't give up what you're doing. Keep working hard on gene therapy, but we have something interesting to pursue. May, we generated cell lines. June, we designed and ordered a series of oligo drugs. In July, we began to have our first careful conversations with the FDA. Are we going to have a route to actually give this? In August, we got our assay set up. In September, we were seeing that the RNA splicing was being improved. In October, we were seeing that the cells were looking happier. Now, to explain this in simpler form, I have a very short video that we'll see if we can play.
And this is a lysosome, which breaks down the cell's waste. But in Mila's cells, the lysosomes aren't working, and they start to fill up. The cell tries making additional lysosomes, but it eventually becomes overwhelmed and dies. It turns out the culprit is a mutation buried deep within Mila's DNA that affects the lysosome proteins. To make a protein, the cell uses its DNA as a blueprint to make RNA by splicing together the essential bits of code. The cell machinery then reads the RNA to build the protein. But in Mila's RNA, a misplaced piece of genetic code has thrown off the splicing process. So when the cell's machinery comes to assemble the protein, it's halted by the misplaced code and the produced protein is defective. Thus, Mila's lysosomes can't function properly. Researchers at Boston Children's Hospital have designed a special drug just for Mila. It's called Milasen. The drug hides the mutation in Mila's RNA with a new piece of genetic code. This allows the RNA to be made correctly, which lets the cell produce normal proteins. The lysosomes can now take out the trash, and Mila's cells stay healthy.
So at this point, it was October of 2017. It was 10 months after Mila had been diagnosed. And she had lost a lot during those 10 months. She had been able to take steps by herself, and at this point, she was needing full assistance. She had been able to speak very slurred sentences, and she was down to saying mommy and one other word. She was no longer able to eat food cut up into small pieces. She was having to eat pureed food. And I was on Facebook seeing other families with her type of Batten disease, with children even just 1 year older than Mila who were in wheelchairs. They were not really responsive. They were being lifted in and out of their beds and their cars. And I realized that time was ticking, and our gene therapy was clearly, it was very exciting for future families, and I'm still involved in that, but it was definitely not going to be in time for Mila. So when Tim called me and said that they had shown a proof of concept and they had shown to restore function to Mila's cells, I couldn't believe it. I had protected myself, my emotions, and even though I was getting really excited about, I was talking to Tim on practically a daily basis for this entire year, I still was very nervous that they wouldn't be able to prove that it would actually work. So when he said that it was, and that they had done many tests in many labs, and he said, I think it's time for you to come to Boston, I think I had to stop and think, what does that mean? And what it meant was that they needed to meet Mila in person. I didn't realize that ethically they needed to make sure that we as a family were prepared for what we were possibly going to undergo if there was a treatment, and whether or not Mila was in the right place to be treated. And so this to me was really amazing. And then at that point, it was a race against time, where both Tim and I knew that we were getting close, but there was a lot more work to be done. And so the clock started kind of ticking at that point.
There definitely was a lot more work to be done. And as Julie and Alec described to us in that October meeting, Mila had a real downhill course, even over the 10 months that we were working. And we actually asked Julia to write a letter that we could share with the FDA, explaining the urgency of the situation, explaining how from January 2017 to August 2017, she was showing concrete declines in all of these different domains. And we needed that letter because we knew there was so much work more to be done. We had really, on one hand, we're proud that we had gotten where we were so quickly, but this was just really the very first step. This is proof of concept. And for typical drug development, there's manufacturing, there's release testing, there's formulation, animal toxicity studies, phase 1, 2, 3 studies, necessary to get something into a patient. And you've all seen the statistics for how long this normally takes. And we thought to ourselves, how are we going to accelerate this? How could we possibly make this work pay off for Mila in a time frame that's going to be meaningful? And one possibility that occurred to us after consultation with lots of folks who had lots of more regulatory experience than me, I had none, was this path called the Individual Patient Expanded Access. And this is an investigational new drug application that is typically used if you have a patient who has a terrible disease that requires treatment, but an FDA-approved existing drug has not been approved for that indication. And the trouble was that there was actually no precedent for using this particular route for something that wasn't off the shelf, for something that had never been in an animal, much less a patient, not for something that was an entirely new drug. But we began to make lots and lots of phone calls. And we hit upon a number of many key advisors, more than I can I'll put them all on the slide at the end. But I wanted to highlight a couple of the key regulatory advisors that helped us in these early stages think through this problem. And Lauren Black, Mary Pendergast, Sarah Goldkind, all folks who had had decades of experience at the FDA, were now off on their own outside of the FDA. And they really walked us through this process. We also had help from Art Krieg, who you all know and will be introduced shortly, who was the president of the Oligonucleotide Therapeutics Society. And I remember a call with Art in October where he told me that in 2 weeks, as we were wrestling with this problem, there was going to be a meeting in Bethesda with industry, academics, and regulators all present. And Art invited me to come share this case and present it as a first dry run to many of the folks in the audience who would actually end up being our FDA reviewers. And with lots of help like that, we were able to come up with a plan to meet the challenges. Now, there were very, very many challenges, and here's just an example of one of them. So there was really no applicable FDA guidance for situations like this. Just take manufacturing. Making the drug quickly and safely was the most critical step. This is a drug that we're injecting it into the spinal cord, and there's very little immune surveillance in that partition of the body, in that compartment. And so we needed a very small batch, only 10 grams, which was about 50 times less than the companies were typically able to produce when we called them. We needed it yesterday, and it had to be super sterile for CSF injection. And the quality guidelines that were published by the FDA simply just did not exist for clean manufacturing at this scale and speed. Nonetheless, with lots of help, we put together a plan to address each of these one by one. And we came out with a very aggressive timeline where we were hoping to get from our proof of concept and clinical evaluation stage through manufacturing, release testing, animal studies, and into a patient trial in only 4 months. As you can tell, we were naive and we had no business trying to do this, but sometimes my old advisor always told me, sometimes going into a field not knowing too much can be actually extremely helpful. And we actually were making really good progress. Manufacturing, as it turned out, was completed on December 12, 2017, with the help of many people on the slide listed here. We made 18 grams, and all of the release tests and the quality testing were within the specifications that our consultants had taught us would be required in order to administer this drug. Now, this was the first step, the manufacturing. There's actually more to the story, not all of which I can fully share. It was actually quite a battle to do this. There's one piece that I will share with you. December 12th was the date it was completed, but we had a hiccup. And it was made, it was sitting on the bench, and it was ready to be shipped, and we got a phone call saying, we have a problem. The labeling. And I said, what's wrong with the labeling? Well, we had agreed that this would be released to us under the term for research use only. But on December 13th, the lawyers called, and they said, we're not going to release it to you under those terms. We want to call it not for clinical use, because they had made it so fast and they had skipped some of the usual safety steps that GMP manufacturing typically requires. And we said, that doesn't make any sense. You know exactly what we're going to do with this. I can't call it not for clinical use. So what about investigational use only? Right? Seems reasonable. They didn't like that. They said, well, we want to call it research grade. And I said, oh, come on. Like, time is ticking. We have a schedule to keep. You know exactly what we're doing for it. How about we link it to our investigational new drug application? Why don't we call it investigational use limited to IND 136294? Or heck, why don't we just label it for Mila? You know exactly what we're going to do with it. And they eventually relented, and this was the compromise. Research grade material limited by US law to intended use as stated in IND 136294. Each vial came with this. It barely fit on the label. But finally, after this last 11th hour negotiation, on December 18th, 2017, 6 days later, they released the lot. And I said, I'm not going to believe it. I want a picture of it. And so here is actually the FedEx box loading into the truck and departing the loading dock on its way to Boston. So this actually had set us back a bit. We had to scramble to rearrange our next plans in our very tight schedule. But we managed to do that. And the very next day, this arrived in Montreal. And we started intrathecal injections in rats. We dosed them at 3 different levels, low, medium, and high. And we followed neurologic exams and histopathology to prove to ourselves that this wasn't going to be poisonous. And prior to our first dose in our patient, in Mila, we negotiated with the FDA that we were only going to look at the results from a single dose in rats followed out to 8 days, because there was no time to wait longer than that. We did have follow-up reports with multiple doses that we continued to send to the FDA, but we wanted to get started post-haste. We developed a clinical protocol, and with my clinical colleagues, especially Chuck Birdie, we developed a protocol to give this by spinal tap. We would do a combined phase 1 and phase 2 trial where we'd be a dose escalation every 2 weeks. And then after we got up to our target dose, we could stretch it out to maintenance dosing every couple of months. And we developed a safety monitoring protocol and a series of exploratory outcome measures that we hope to use to tell if the drug was working. We invited Julia and Alec, and it was a happy day on January 13th when they actually arrived in Boston as all of this was going on. Now, I have to admit, as happy as we were and excited as we were to see them, we actually hadn't received permission to proceed yet. That came 6 days later. We got a letter from the FDA after a final review of our early rat data that we had permission to proceed as planned. But we actually hadn't received the drug yet. The drug was still actually in quarantine, finishing the last rounds of release testing. But on January 25th, these precious bottles, these precious vials, arrived at Boston Children's, and we shelved them away in 2 different freezers, one on the 6th floor, one in the sub-basement, on 2 different circuits to make sure that nothing would happen to this precious supply. And on January 31st, after taking a deep breath and one last look around, we launched our trial.
At this point, it had been such a race that it felt obviously very surreal that we were at the point where we had moved to Boston for we didn't know how many months at that point. And I hadn't had any time to stop and actually think about what was about to happen. I did know that I had no hesitation. I knew what was going to happen to my daughter. She did not receive this treatment, so I never stopped to actually think about it. But the night before, I did go on a run in the absolutely freezing Boston January, 7 feet of snow that just kept accumulating over the, few days that I was there. Day and just sat and I had a thought and I couldn't believe that it was actually happening. I couldn't believe that 1 year before, which in the world of rare disease is unheard of. It's a miracle if any child with a rare disease has a treatment available to them, and usually parents that fight take years and millions of dollars, and very often it's not in time for them. So the fact that it had been 1 year since I started speaking with Tim about just looking for a missing mutation, and here I was in Boston with my daughter the next morning going to be receiving a first-in-the-world genetic treatment that could offer her hope, seemed really unbelievable. Leading up to this over that year, I had started a website for our foundation, Mila's Miracle Foundation, and I had written a blog that just really was an outlet for me to write when I felt inspired. And that night before her treatment, I wrote a blog, and I just took a short little excerpt, which I just thought I would read. I entitled it Between Hope's Edge and a Miracle. On the eve of Mila's treatment, I'm filled with many emotions. I've pushed so hard over the past year to get to this point that there was little room left to think or feel. Now I'm here sitting on the hardwood floor of our rental house in Boston with my back against the wall. Mila's treatment is scheduled for 12 hours from now. I hear myself breathing. I feel my chest expanding and collapsing. I look around the room and seem to notice every detail, perhaps because this is the first time I've allowed my eyes to drift in over 1 year. Part of me just wants to hit the fast forward button and have Mila's first treatment behind us, but something pulls me back. Deep down, I know that I am in a sweet spot that I may never be in again, a spot between hope's edge and a miracle. Tomorrow, Mila will receive an injection of a new medicine that will go directly to her brain. It is experimental, which means it could be that breakthrough treatment that makes the impossible become possible, or it could be a valiant attempt that just falls short. She could be the pioneer whose life is saved, or the one whose sacrifice helps those that follow.
So thank you, Julia. So today it's May 2019, and we're about a year and a half in. And I wanted to share with you what we've been doing, how Mila is doing. This is the series of doses that we arranged for her. And beginning in January 31st, we started with 3.5 milligrams, and then we gave her slowly increasing doses up over a series of about 3 months. And we followed her during this time with measures of her strength, with imaging, with EEGs, with neurologic examinations, sensory examinations, drug PK sampling, safety labs, all to ensure that this critical period would remain a period of safety for her. And then since then, we've extended dosing out to maintenance doses approximately every 3 months. So she's been coming back to Boston, flying to visit us, and having another dose. How's she doing? Well, all the caveats that you can imagine have to be applied here. This is an N of 1 treatment. There's no control. There's no placebo. There's no switchover period. There aren't any of the things that clinical trialists typically have to convince themselves then that what they're doing is working, except for patient reports and a couple of key outcome measures that we were hanging our hat on. And I'm very happy to say, first of all, from a safety standpoint, that the drug to this year-and-a-half date has been well tolerated. There was one incidence of a spinal headache on dose 3 or 4 that lasted for a few days and then went away. And there have been no adverse effects that we've been able to follow in any of our monitoring to date. Now, with respect to outcomes, we've been collecting seizure logs and video EEGs and neuropsychological measures, but I'd like to invite Julia up just to explain what she's been seeing, and then we'll go through some of the data.
So right before Mila's treatment, unfortunately, that same time that the labeling issue happened, while that was funny to watch here, it was not so funny for us, as every day really mattered. Mila, Batten disease, and a lot of neurological conditions kind of are marked by maybe a slower regression and then a cliff, and a slower kind of regression, a cliff. I was hoping that Mila did not go off a cliff before her treatment, but she did, and it happened at the same time as this labeling was going on. So it was a pretty horrible December and January, knowing that the treatment was on the horizon, and Mila was suddenly losing everything, like by the day. She had gotten to the point where she was fully slumped. She was choking and coughing on her pureed foods. We had to put a G tube in her, which started out with water and very soon after became a way of supplementing formula and food. So she was eating less and less by mouth, receiving more by her G tube. She's standing, she was still taking some steps, but I was having to hold her up. My arms were so sore every day. My back was sore, my neck was sore. When I would sit her down, she would just kind of like flop over. Her seizures, which is very typical of Batten disease, had been, I didn't notice them at all until about November of 2017. And suddenly they shot up by the day, and they got to the point where the week before her treatment, they were at 30 a day. They were kind of seemingly cute because she laughed, gelastic seizures, which are quite unusual. They were starting with laughing, but every week they got worse and worse, to the point where I was having to hold her down physically at the dinner table, breakfast table, lunch table, whatever, all the time. And they were lasting 2 minutes. And she was arms and legs thrashing and kicking and smashing her legs up against her tray. And I was barely able to hold her on her chair. And so all of this just suddenly happened in those 2 months leading up to the treatment. Now, zoom ahead, not too far, maybe a month or 2 or 2.5 months after the treatment. As you saw, Tim was saying that they had to dose escalate. So it took quite a while for the drug to kick in and to have enough in her system to make a difference. And suddenly, she was not having those seizures anymore. She was having much less severe. She was no longer moving her arms and her hands. They were going down in terms of number per day. And within a few months of her treatment, she was having somewhere between 2, 3, 4, and maybe 12 in a day, but they were lasting a few seconds. And if anyone has any experience with seizures, they can be very, very disruptive. They can be all day and just kind of make the whole day difficult. And suddenly it felt like Mila really wasn't even having seizures. If you had walked in her house, and still to this day, you probably wouldn't even notice she's having them. They, she laughs and moves her head for a few seconds and then kind of goes away. We've had 1 or 2 in the last week or so, and we've had 10 one day, but they're so minor. It's just an incredible difference in quality of life for her and for all of us. She still, everything with all of these diseases is, every day is different. So there are days that she has that are great and other days that are not as good. In general, her posture has been much noticeably better. So she can stand against me like this while I grab things, and she will fall over if I don't hold her, but her upper body strength is just so strong, and I can sit her down and she will not crumple. She might fall sideways if I don't put something next to her. It's not perfect, but she's able to hold her neck and her head and her torso up. It means that when I'm holding from behind and she's walking, her body is up so she can take steps as opposed to being fully slumped. We no longer give her any supplemental food through her G-tube. She receives water because it's very hard for her to suck through a straw. But she's eating pureed food fully by mouth now. So once again, just a huge difference in quality of life. And what's most important for me is that the month leading up to the treatment, she was losing who she was. It was the worst thing that I've ever experienced in my life. It's worse than the thought of losing my daughter, was seeing my extremely outgoing, just laughing, smiling daughter not respond to the songs and the books that usually made her laugh. And that was happening in the 4 weeks probably leading up to the treatment. And I started noticing a few months into the treatment that it's not black and white, and it is not today either, but definitely I see much more response to the things that made her laugh before and the things that made her smile. And she was listening carefully, and when her brother would do, crazy, loud, funny things, she would laugh again. And once again, every day is really different, and every part of the day is different. But just the fact that she's here in the world and paying attention and laughing and listening and responding is probably the most important thing for me. So, all of these things have shown that, our hope in rare disease right now, the name of the game is it's a miracle if you can stop a disease. No one is pretending to cure, a rare disease right now. Stopping it is amazing. Mila's rapid decline from this treatment seems to have stopped. It is too early to know whether or not it has stopped or it's on a very slow decline, which is also possible. It's only been a year and a few months. But there's a very good reason to believe that it's working very well. And obviously, time will tell. But for right now, there's a lot of good reason to believe that it's working as well as Tim and his team had hoped it would.
So as you can imagine, thank you, Julia. As you can imagine, we've been following Julia and the family's reports very, very carefully. These patient-reported outcomes and quality of life are really what it's all about when you've only got a single patient. We have been also collecting the measures, the objective measures that we can. And we have a series of neuropsychological assessments, mainly based off of the Bayley scales for infant development, Vineland scores for adaptive behavior. And I'm happy to say these are noisy scales, and they're not that great for kids as sick as Mila. But they do seem to corroborate what mom and dad are telling us, that her steep decline that we'd seen in the months going in has seemed to have leveled off. From a seizure standpoint, this is perhaps the piece that gives us the most optimism. This is a chart of her seizure trends during the first 4 months of treatment. And what you can see is that from this high of about 30 seizures per day that Julia was describing, each of them lasting over a minute, sometimes up to 2 minutes, from the dark circles here. As the doses in the orange asterisks go up, we see this really remarkable trend where the frequency and the intensity seems to be going down. Now, after this escalation, we began spacing the doses out further. You can see the data gets a little bit noisier. Here, the initial trend is still clear, but now we're perhaps getting some rebound. However, we take a lot of solace in the fact that the seizure intensity remains consistently, chronically suppressed since we started this. And we continue to follow this month by month, EEGs. We also have video documentation and other electrographic confirmation of the daily seizure reports, and we'll continue to study Mila and see how she does. So I'm going to wrap up with a few concluding thoughts, and then we're going to open up for a discussion. But just to summarize, it's been a remarkable privilege to be involved in a project where we have surprisingly found ourselves in 1 year going from a diagnosis to a personalized medicine. This is an individualized drug. Mila's mutation that we found in her has only been found in Mila. We haven't found it in anybody else. We've screened through so far about 500 whole genomes and don't see it at an appreciable carrier frequency. Now, I think, as Mark pointed out yesterday, when you find a diagnosis for a rare disease, a new gene, and you have a treatment, more cases may spring up. Maybe we'll find more cases in the future, but for now, Mila is the only one. Still, even as a drug for one patient, I think it has some really interesting implications, and I'm going to challenge the audience to think through those with us in the ensuing discussion. It was critical to have close, tight collaboration. Julia and I spoke on the phone with her scientific advisors for her foundation nearly daily for almost 6 months. And I was similarly on phone calls and texts with folks in industry, our FDA, ex-FDA consultants, and CROs that all banded together to make this happen. And it takes a lot of real tight collaboration and openness to this idea for someone to risk their pharmaceutical platform in a way like this. It does raise, however, a very interesting future possibility. Could one imagine having someday rapid genetic therapy clinics for children with fatal conditions like this, where could you turn this process into something that's repeatable and scalable? And if you think, initially you think that's not going to be possible. How could we possibly do this? This took so much effort. But let's explore that a little bit afterwards. I just want to show us a few slides showing representative photos of the teams that assembled to help Mila. And this is a shot of the TriLink Biotechnologies, which really banded together. They had penny jars and donations and Mila's pictures up on the wall as they were busy making Mila's drug. This is a picture of Brammer Bio in their Florida facility. And this team took the powder that we had made and filled it into sterile vials ready for us to administer into our patient. This is Chuck Birdie, an anesthesiologist at Boston Children's who has had decades of experience in investigational intrathecally delivered analgesics. And it was his experience having filed INDs in the past that allowed me to partner with him and make this happen. And this is the huge team from many other walks of industry, academia, regulatory science, funding, acknowledgments, that it really takes a village to pull this off. The question is, can we do it again? So with that, I want to say thank you to Julia.
That was great.
Wow. No matter how many times I see you speak, it's still extraordinary. So what we'd like to do next is have Art Krieg come up to moderate a discussion with Tim and Julia. Art has actually done much of the foundational work in this area, actually, as it turns out. And the one thing I noted in your bio is you have an H-index of 125 with 250 papers. So I guess 125 are good and 125 are just okay.
It includes patents.
Oh, okay. Well, there you go. So, Art.
Thanks very much, Matt. I mean, what an incredible story. I think it's just amazing. It's a proof of concept. This is something that has never been done before, but it can be done again. And so what I wanted to do is to just talk a little bit about why it can be done again and how it can be done again and where the field is at for therapeutics. So as you've heard before, developing one new drug classically has taken $2.5 billion, more than $2.5 billion, more than 10 years. And the reason is because in the past, you started out with basic research and drug discovery where you're screening thousands of different molecules that could become a drug, and you're weaning them down to come up with one molecule that is a drug. And you then go through these clinical trials and so forth. That process obviously isn't going to work for rapidly progressive neurologic disease. And so how can we do better? Well, The reason that drug development in the past was so expensive is because historically the vast majority of drugs have been small molecules. And these are numbers from when I was at Pfizer. These were our internal metrics for starting a new drug development program on a new target. How many people would we have to put on that? How much was it going to cost? And how long was it going to take? And on average, over hundreds of different programs for different targets, you're talking a lot of people, a lot of time, and it's relatively expensive just to get to a lead compound. And this is a compound that's ready to go into animal testing and go through all of those other steps. So this obviously is not something that's going to be amenable to treating a rare disease and to moving quickly. Also, for big companies, it makes them relatively conservative about taking on new projects because it's a big commitment to work on a new target. And so you try to kill new projects early before you've spent too much money on them. Now, antibody therapeutics have come along in the last decades, and they're cheaper and faster. It takes fewer people, less time, and it's less expensive now to get to a lead compound. And I just wanted to share with you a little bit about how the antibody platforms got to where they are and the steps that had to occur for us today to have many biologic therapeutics. The original discovery that made that possible was in 1975, the discovery Kohler and Milstein, how to produce these monoclonal antibodies. The first one that was actually approved was in 1986. This was a mouse antibody. That was the original approach to making antibodies. It was not a commercial success. People make antibodies against mouse antibodies that limit their efficacy. A lot of innovations had to occur in the following decade. Many companies in the field went out of business because they weren't able to get anything commercialized. A lot of big companies gave up on it. Finally, the first commercially successful antibody was approved in 97. And now this is a platform that can be used for drug development. Every major pharmaceutical company is cranking these out. It's become industrialized. It's still not really practical, though, for rare diseases. It's too slow for N-of-1 therapeutics, and it's pretty expensive. Nucleic acid drugs are a game changer for the field, and you heard a little bit about that already. We estimated at Pfizer, they had never been done before, but we estimated that we ought to be able to develop nucleic acid drugs with fewer than 10 FTEs in less than half a year to get to something that could go into animal studies, and for just a few $1 million. That's Pfizer metrics. Pfizer has a lot of bells and whistles. You could do it in an academic lab cheaper and faster. I don't know, I mean, it's amazing to me how fast Tim and his group did it. And one of the things that's amazing too, I had never heard of Tim before. He had never worked in oligonucleotide therapeutics. And to me, this is one of the other amazing things about this platform that shows that someone who's never done this before can come in and talk to other people and make it happen and make it succeed. This platform has enormous potential. It's a really exciting platform that can be applied for many other diseases. Not every disease, but it's got huge promise. This is rational drug design. I hesitate to use that phrase, but the idea here is that because we know the sequence of a gene and we know the sequence of an RNA, we can design an oligonucleotide with the sequence that's going to match that. And so instead of having to screen through thousands of different compounds and go through many generations of antibodies, you have your sequence, you make your drug, you make several because you have to experiment a little with them, but it's a much faster, simpler process. The same basic chemical building blocks are used for all of the drugs. And they don't vary that much in terms of their toxicities, the off-target effects. And so once you've characterized the pharmacokinetics and the basic properties of one, once you've worked out the manufacturing for one, you've got it for all of them. And so it makes it possible for us now to scale this up. The cost is now in range for N-of-1 therapeutic development. So this is sort of what we're talking about. Traditionally, drugs targeted proteins, small molecule drugs targeted proteins. But proteins are made by RNA and by DNA. And so now we're targeting at the RNA level upstream from the proteins to fix the proteins before they actually can cause the disease. So the mutations, of course, are in the DNA. That's what's causing the disease. Conventional drugs work at the protein level. And where the whole antisense field started, I guess I can go back even further. In 1978, Paul Zemechnik, a very smart scientist, came up with this this idea. And at that time, it took 6 months for a graduate student to make enough ASOs for one experiment, and that's just an experiment in cells. And actually, the chemistry that he used could not possibly have worked. So he published this in PNAS, but it actually couldn't have worked. It was a great idea. And then in the 1980s, when Marv invented automated DNA synthesis, all of a sudden people realized, wow, now we can come back to this and we can scale this up. Also, there were new chemistries that were developed during that time. In 1993, Richard Cole first conceived of the idea to alter RNA splicing using oligonucleotides. He told me when he submitted his manuscript on this idea for publication, it was rejected. One of the reviewers called it molecular gymnastics with no conceivable practical value. But at the time, actually, it didn't work in vivo because the chemistries still were not good enough. They didn't bind tightly enough. They weren't stable enough to be effective. And although there was an ASO drug that was approved in 1998, it actually didn't function as ASO. It was based on an early chemistry that had a lot of off-target effects. It required, again, generations of innovations, new chemistries, new designs. Literally billions of dollars were invested in this field, and many of the investors lost every bit that they put into that field before finally, as you've heard, Adrian Craner coming, and then finally nusinersen being approved by the FDA to treat spinal muscular atrophy. This was the first commercial success for the ASO field. This is proof of concept, and the same basic approach, as you've just heard from Tim Yu, can now be applied to a whole lot of other diseases and conditions. Not everybody, but that's what we want to talk about now. Where do we go from here? What's next? And so I'd kind of like to open it up to people. I've got plenty of questions I can ask Tim and Julia too, but I'd like to give you all a chance to ask questions as well. So well, I'll just bring you can bring in the microphone.
Thank you. I don't mean to be intentionally simple-minded. Why did you need FDA approval for this?
This is a space where I have argued in the past that an intervention like this could be considered a procedure, not unlike a surgeon going in and doing an emergency appendectomy. But that argument didn't really fly. We were actually working with companies that were making the drugs for us on a contract basis, and the opening of a clinical trial in a situation like this is regulated by the FDA. And there are arguments about going back to the original charter of the FDA and their role to regulate interstate commerce. There are arguments that you can make that this is not a commercial drug. This is something, again, for a single patient. But we didn't want to risk upsetting the apple cart. We'd rather get their buy-in on this than try and do an end around.
And Tim, could you characterize, too, the FDA response? Has the FDA been obstructionist? Have they been bureaucratic?
So we went in with a lot of trepidation, actually. We were worried that this was going to completely blow their minds and that they would not allow this. But actually, they have been incredibly supportive. And I think from the initial call, when I literally just looked them up on the webpage and I called the 1-800 number for emergency INDs. And they said, have you run a clinical trial before? I said, no. But from that initial interaction, when they came back with a list of requirements that were initially too long for practical use for Mila, we engaged in a conversation. And they were incredibly supportive and continue to be incredibly supportive of this concept. Julie and I actually just came back from a meeting with them where the two of us explained what we did with the Mila case. We thanked the Division of Gastroenterology and Inborn Errors that were our partners throughout all of this, and had a conversation with them about their interest in seeing more of this happen. And I think that we can say that we came away very, very positive, that they see this as something that has to be dealt with, and we're looking forward to working with them.
Hi, my name is Amber Freed, and my son has a rare neurological disease called SLC6A1. We're developing a gene therapy along with the University of Texas Southwestern, and I aspire to be sitting there like you guys someday. Poor Julia responds to a lot of 2 a.m. texts from me, and she's always so sweet. What advice would you give families for in positions like Julia right now?
So I think it's a pretty remarkable time to be working in this space. I think that there are many examples of citizen science that have really blown my mind. And I've been very, very fortunate to partner with Julia in this incredible adventure. And there are many more stories like this that are coming out, and I hope yours will be soon one of them too. I think that not being afraid to talk to researchers who are really jumping at the opportunity to work with families. For many folks, the diseases that we work on may actually be quite abstract. Not everybody is quite so lucky as to be able to see patients and also work on those patients' diseases. That flip between physician and scientist is not something that everyone has. And if you don't have it, you're actually giving people the opportunity to engage in that. And I think that don't undervalue what you bring to reaching out to folks.
And Julia, how would you answer that question?
I struggle with the answer to this question often because I know that it's not easy to do what I did. I raised $3 million in 1 year, not of my own money. And I didn't sleep for a year. People ask me all the time how I did it. I don't know the answer. I just saw my daughter who was dying, still smiling and laughing, and I just did everything I possibly could. So I know it's not easy to just tell another parent, just do it, because it's very, very difficult, and especially difficult right now because now gene therapy and other therapies are becoming more common, and so there's a lot more kind of competition for raising money and getting interest from scientists and clinicians. I just think that, we're a giant army here and that the average person doesn't realize that while Mila's story seems so incredibly devastating and impossible, there are millions of Milas in the world, unfortunately, with just genetic fatal diseases. And as parents together in the last 2 years, it's changed so much in terms of the parent's voice and how important it is with the FDA and with pharmaceutical companies. Rare disease was nothing 2 years ago, and now companies are scooping up the IP for ultra rare, gene therapies, and they're spending tens of millions of dollars on them. So there is now a value for ultra rare diseases. So I just think it's unfortunately a team effort that may or may not be in time for any of our children as kind of the pioneers. We hope it is, but we know that together it's going to ultimately pave a path for many other rare diseases that follow.
That's a wonderful story. I mean, an unfinished but wonderful story. I've spent a number of the last few years working on accelerated access to medicines, not these medicines, but other medicines. It seems to me back to the first question that was asked in the relationship with the regulators. I've found regulators actually quite understanding if you engage and you engage early. And I think, as you were just indicating, this is the beginning of a wave of therapies. They won't all be antisense therapies. They don't always work. There'll be gene therapies, gene replacement therapies, gene editing therapies. And I think what you've done is done a pathfinder project, in a way, for what we have to have work. Because unless we can do these fast and we can do these cost effectively, these will be driven offshore. We already see examples of that. So I think it's in the interest of the FDA, in the interest of patients, in the interest of the people who are going to commercialize these products, they won't all be commercial in that full sense of the word, to create a new process. And I think you've laid out wonderfully the elements of that new process. And I think there's a generalization that needs to be done here. Before, as I say, some people go to places, the Dominican Republic or wherever it is, and get this done unprofessionally. We need to build a system that takes the lessons that you've so wonderfully laid out and make them more generalizable. I mean, sorry, it's not a question, it's a kind of comment.
That's a wonderful comment, thank you.
So I teach the undergraduate molecular biology course at my institution, and I use Adrian Krainer's work with my students, and they read his primary papers to sort of help them understand splicing and translation by the ribosome. And the reason I do this is that most of my students are pre-med, and I want them to understand that the landscape that they're going into is so different than what it would have been 20 years earlier. Medicine is just revolutionizing, and the story of nusinersen is, a great example of that. But I actually had a question for you. So, because I've had to read these papers so carefully for my class. So Adrian tested so many different ASO variants, and you didn't have time to do as much. There will always be an ASO that's a little better at giving you more of the splice variant that you want, and that is going to function in an improved way. Through this process, is there any mechanism for you to adapt your ASO and create a new one? And will the FDA enable you to implement it with Mila?
That's a great question. So you're exactly right. In the development of the drug for spinal muscular atrophy, hundreds of compounds were tested. And in fact, they just carpet bombed the entire stretch of RNA nearby the abnormal splice event with hundreds of antisense oligonucleotides, each differing from its neighbor by only 2 or 3 bases. And right, we had no resources or time or hands to do that. We tested 6. And we did this because we had computational resources to predict where the best spots ought to be. Once we hit those 6, we had to weigh perfect being the enemy of the good, time versus optimization. And our particular strategy here, the tactic, was to proceed by taking our lead candidate that we had hot in our hands, get it into rats. In the meantime, our postdoctoral fellow was scurrying and making 20 different variants of that initial lead. And we had them as backup in case anything went wrong with the first rat. And that's something that we would advocate doing if there were future cases. In some cases we're prosecuting now. And we would bring those back to the FDA if there were the need to do that for Mila.
Back here. I wonder if you've had any discussions with NIH about creating a fund to support some of this, making many more ASOs and doing this kind of thing. Because, I mean, Ionis is great to work with. I've had 2 ASOs from them, and they're wonderful. But it's because they were interested, and they said, "Okay, we'll do it," and they pay for it. But if they didn't, it would be too expensive, and it takes time. And so I wonder if NIH would be able to get behind any of these, ASO projects to get them made, and then at the same time, when there's a new disease that you want to work on, paying for the CRISPR mouse to just get that made as quickly as possible, get the ASO made as quickly as possible. Have you discussed this with Francis Collins or anybody with authority at NIH?
Thank you. Yes, we have. So we've talked to Francis Collins about this, and there's a lot of interest at the NIH in this concept. Julia and I, we've really partnered together in our approaches. I think Julia brings the incredibly powerful patient voice. Her foundation comes in representing just what patients want. I wear my Boston Children's hat when I approach folks, and we represent the academic medical center angle. And there's a lot of interest in the NIH on platform technologies. We've been talking to the Center for Advancing Translational Science, Chris Austin and his team, and the Office of Rare Disease. And we're in discussions with them. I think that the concept that this is a perfect type of project for our government-sponsored institutions to be working on. And the idea of creating a platform upon which many patients can benefit without cordoning it off into too tightly held IP held by one particular entity. I think that's what they really see the value. They see this as really aligned with what their mission is. we hope to do things with them too.
Okay. So you learned a lot about Batten's disease, with this particular single case, and the drug seems to be safe and well tolerated, and he seems to be observing some benefit already. Are you thinking of doing maybe additional patients to give more validity to this strategy?
Would this be a good time for, so Julia and I did prepare a couple of slides to share with folks what we see as immediate next steps. And maybe perhaps here's a time where I might share those with you. So we do think that there is an opportunity to keep trying this. And we do need more data. We have a single case so far, and we have to prove that, one, it's possible to do again, and that, two, it could be potentially safe and efficacious again. So we did prepare a few slides to share with you on this. So what's next? We first started talking, our first time speaking about this publicly was in October of last year. And it's been picked up by a couple of news outlets. We have a manuscript under review. But even with just this first conference presentation, there's broad interest. So there's a lot of pressure on us to figure this out. We've had over 50 requests from clinicians, families, patient organizations. My lab in particular has screened through several hundred different genetic reports from patients. We're looking for other opportunities to try this type of intervention. And other folks in the space are jumping into the breach to fill this need too. Right? And we personally have identified to date over a dozen families that have mutations like Mila's that we want to try to help. This slide just sort of, I think we've talked about this, the idea that this is a potential platform and that it's, in a way, sort of a programmable medicine. This is nusinersen for SMA, this is Mila's drug. And just by changing the letters but keeping the chemistry the same, we ought to be able to gain experience with what that does from a pharmacology standpoint. So far, the pharmacology, the half-life, the toxicology, the biodistribution, they share a lot if you keep the chemistry the same. And so we've been thinking, well, what are the definitions of cases like Mila's? What was unique about Mila's case that allowed everyone to so quickly agree that it was appropriate to do this? And there were a couple of things. These are terrible disorders. They're progressive, they're fatal, they're life-threatening, and so the risks of trying something extraordinary like this are balanced against the risk of the natural course of the disease. Two, these are identified single gene mutations with scientifically feasible fixes. This is easier than neural tube defects. This is easier than diabetes. These are single gene disorders presenting in childhood when fewer other things have gone wrong. And so this boosts the odds that a genetically targeted treatment could work. And then last, these are generally come in very small packages, n's of ones or very, very small numbers of patients. And so from the standpoint of a public health risk and the FDA's mandate to protect the public, we're exposing individual patients, talking very carefully to families that are capable of understanding how complicated this is, and consenting them one at a time. So this is not so much a public health issue. So we presented these criteria to the FDA, along with the idea that there are a lot of people who could actually fit and meet these criteria. This is back of the envelope calculations, hasn't been validated. This is just literally in my office, counting through the numbers of rare disease individuals in the US, the numbers that are genetic, the numbers with neurologic diseases, and a third of these are fatal. And there are 2.5 million patients with a fatal diagnosis right now. And if 10% of them are treatable like Mila's, if they have a mutation that makes it amenable, this is potentially, there are many individuals with whom we would have the opportunity to work to try to explore this. So Julie and I presented this white paper, which is a little bit of a manifesto describing why we think further exploration with patients meeting these criteria is warranted. And the key elements are, one, the idea is that we want to do a pilot of around 12 more patients like this with meeting these criteria. And this will let us test the safety, the tolerability, the efficacy of this approach. The second goal, besides repeatability, is that we have to see if this is actually can be made to be accessible. And here the idea is that if a data set of 12 shows that this is actually safe and scientifically and clinically sound, then we have to find ways of making this as accessible as possible. And Art explained all the advantages that ASOs have, ease of manufacturing. It's got decades-long stability. In a single manufacturing run, we could make hopefully a lifetime supply for Mila. So developing the methods to accelerate every step of this process to make it reach out to as many patients as possible, that's got to be the other goal of this. But first, we have to show that it's safe.
So I'm wondering about range of action. So it's really wonderful, but the brain is very deep. The brain is deep and layered. And so I'm wondering about how far the ASO is penetrating through the brain tissue and that get to reach the cells that it needs to. And is that kind of the next future step that we need to be thinking about, as well as kind of how do we get it to the cell types that are needed?
That's a great question, Lee. Art could speak to this as well with his long experience with antisense oligos, but before we looked into, as we were looking into the feasibility for Mila, we looked at the biodistribution studies that folks like Frank Bennett and Frank Rigo from IONIS have published, and others as well. And there have been some beautiful nuclear imaging studies, as well as careful sectioning studies that show that when you inject ASOs into the spinal cord, it penetrates frontal lobe, cerebellum, the motor cortex, multiple regions, including some penetration to deep nuclei as well. It's lower in deep nuclei like basal ganglia and so forth, but sufficient that we felt that this really could possibly work. So more data like that needs to be generated and or released to the public domain. And there are also human autopsy specimens from children who are being successfully treated with Spinraza but may have died of other causes. And that type of data that we gather over the next few years will kind of help build the case.
Okay, should we just do one more question? I know we're running out of time. Okay. Oh, yeah, sorry, go ahead, yeah. Should we make that last question?
Drug companies are often quite keen on enforcing intellectual property they believe they have. And it sounds like you've copied a huge chunk of a drug that's on the market for an N=1 application. How do you navigate the potential problems if actually you try and scale that out to the N=240,000 that you suggest? Are you going to run into IP problems?
So I think this is a good opportunity for me to actually publicly thank, there are many scientists at IONIS, including Frank Bennett, who were very generous of their time talking to me. They couldn't give me data, they couldn't share compounds or trade secrets, but they were very generous and willing to give of their general advice as we were navigating this, and so I want to say that publicly. We were a little bit shy about saying it early in the FDA process because we didn't want the FDA to be concerned that this was somehow a commercial end around in some way. So I do think that the IP issues will need to be addressed, but from my standpoint as an academic at an academic medical center, these are not profit-making drugs. And I hope that industry will pledge to give their potentially similar platform technologies give us access to that type of IP for what we think are worthy experiments and compassionate experiments like this. And I actually think it's a win-win. I think that we have families who need something and there's nothing coming. And if in the course of doing cases like this, we can offer hope to families and also learn about general properties of a given pharmaceutical approach, everybody can benefit.
Yeah, this approach is in the clinic now for Huntington's disease, ALS, and there are positive results coming out with those diseases, and I think there's going to be a lot more coming too. But recognizing the time, I guess we should thank Tim and Julia again for an amazing journey.